can join. So I'm very happy that we have another edition of our VAC4EU Journal Club. I would like to thank everybody who is joining here. And also, of course, I would like to thank the organizing committee, Sima, Giorgio, and Jose Martin here from VAC4EU side. And also, of course, I would like to thank very much Samantha for taking this on and having prepared this presentation. So I give over to Sima for the introduction. Thank you very much.
Thanks, Daniel. Hello, everyone. Good afternoon and welcome. It's my pleasure to introduce our speaker, Samantha Duckworth. I will give a brief introduction of her background. Samantha is an epidemiologist at Paraxel in Spain, in Barcelona, and she's a doctoral candidate at Utrecht University. And she has over 5 years of experience in both industry and public health, and she has expertise in drug safety, effectiveness, and risk immunization. Thank you, Samantha, for joining us and accepting our invitation to present today. If you would like to add anything to the introduction, please do, and go ahead with the presentation.
Nothing to add. Thank you for the invitation to be with you guys today. Look forward to this discussion with everyone. So I'll go ahead and dive in. So just a reminder of the learning objectives of this Journal Club session is to understand how target trial emulation can be applied to real-world vaccine safety studies, explore key maternal and infant safety outcomes relevant to RSV vaccination, and finally to discuss pragmatic matching strategies to ensure valid comparisons between the vaccinated and unvaccinated groups. And just a quick check before I go further that the mic is working and everyone can hear me well. Speak now or forever hold your peace. So we're going to go through just follow the structure of the paper, the introductions, methods, and discussion. So this paper was published in March 2025, and it outlines a generic protocol for target trial emulation in maternal RSV vaccination. I think the slides will be circulated after. If you don't already have access to the paper, you should be able to from this link here. And the authors are listed below, and they also include my PhD supervisors, Odette de Bruin and Miriam Sturkenboom, who I know many of you probably recognize. So first, a little background, a brief overview of the burden that respiratory syncytial virus causes. RSV is a major cause of lower respiratory tract infections in young children. In 2019 alone, RSV was responsible for 33 million lower respiratory tract infections globally and led to 3.6 million hospitalizations and around 100,000 deaths. Infants under 6 months are particularly vulnerable. They also face another challenge as they're at an increased risk of vaccine-induced enhanced respiratory disease. So this is why maternal immunization is so important. By vaccinating during pregnancy, we can provide passive immunity by transferring protective antibodies across the placenta to safeguard infants during the critical early moments. And building on the previous point about vaccine-induced enhanced respiratory disease in infants, in the 1960s, researchers tried to immunize children with a formalin inactivated RSV vaccine. Unfortunately, when those children later encountered RSV naturally, their immune response became overly aggressive and it clogged their lungs and caused severe illness and hospitalizations. This setback stalled RSV vaccine development for decades. So yeah, I just wanted to highlight that point that it's an exciting time right now that we have. Just a few years ago in 2023, the first maternal RSV vaccine was approved by the European Medicines Agency, the US Food and Drug Administration, and the UK Medicines and Healthcare Products Regulatory Agency. These approvals were based on evidence from a global randomized double-blind placebo-controlled trial called MATIS. and the trial evaluated the efficacy and safety against RSV-related lower respiratory infections in infants born to their vaccinated healthy women during 24 to 36 weeks of gestation. And we're going to be focusing on this Target trial throughout this presentation when we talk about how we're going to emulate. So why do we need to gather more evidence with real-world data? High-risk pregnancies and immunocompromised women were not included in the original clinical trials. This means we still don't have complete safety data for these groups, so that's where post-authorization safety studies are essential to fill this gap. In addition, Guillain-Barré syndrome is a rare neurological disorder that was actually reported as an adverse event and not in pregnant women, but in other adults in the RSV trials. Therefore, due to its seriousness, it still remains a key safety event of interest. In addition, there is a lot of uncertainty about the risk of preterm birth. There were no differences in the preterm birth rates between the vaccine and placebo groups in the trials. However, another trial with a similar maternal RSV vaccine was terminated early because of a higher risk of preterm birth. So in summary, we need more data for these high-risk groups. We need to assess key safety concerns like Guillain-Barré syndrome, preterm birth, and other safety outcomes that we're going to address in the next upcoming slides. So we'll go through for the methods, the critical outcomes and covariates of interest, the study design and statistical considerations, and then discussion of the methodological strengths and challenges of implementing target trial emulation studies and RSV vaccines, studying RSV vaccines in pregnant women. So outcomes of interest include hypertensive disorders of pregnancy, such as gestational hypertension and preeclampsia. Other outcomes include Guillain-Barré syndrome and preterm birth, as I mentioned in the previous slide, as well as stillbirth, low birth weight, and small for gestational age. Each of these outcomes has its own specific exposure window, which is really important. And this means that the time of the vaccination relative to the gestational age is critical for accurate analysis. If we misclassify this timing, we could introduce immortal time bias, where the vaccination appears more protective because women vaccinated later on had to remain pregnant longer to receive the vaccine. And we're going to continue discussing this theme later on, but the authors really emphasize the importance of precise timing, the meaning between the vaccination and birth rather than a binary variable. And this is critical when designing RSV vaccine safety studies. Now we're going to discuss the covariates of interest. The crucial covariates of interest for matching are gestational age, calendar time, maternal age, as that influences both the exposure and the risk of adverse outcomes, immunocompromised status, and high-risk pregnancy. And we also have variables associated with both outcome and exposure. We see a bit of overlap here. The key confounders include the maternal age, parity, prior adverse pregnancy outcomes, lifestyle factors such as body mass index, smoking, and alcohol use, chronic conditions such as hypertension, diabetes, kidney disease, and autoimmune disorders, as well as multiple gestation, history of infections, medication use during pregnancy. And collecting all of these variables is really important, especially for adjusting for confounding and making valid comparisons between the vaccinated and unvaccinated groups. That's going to be a key theme that comes up in this presentation. So the authors recommend an observational retrospective cohort study comparing pregnant women who received the RSV vaccine to those who do not, or with an active comparator group if one becomes available. Currently, there's only the one vaccine that was approved 2 years ago, Abrisfo. The study is designed to emulate a pragmatic randomized trial to assess the safety of administering a single dose of the RSV vaccine between 24 and 36 weeks of gestation, compared to no vaccine during vaccination during pregnancy. Of course, we can't truly randomize the participants, so we emulate the future by carefully addressing confounding biases and using techniques like matching and careful inclusion criteria. And finally, the time zero marks the point when all eligibility criteria are met and when the vaccine is administered. Oh, I see somebody has a hand
Yes,
up.
Samantha, can you go back to the previous?
Yes.
So vaccine randomization, now I'm a bit, so confounding is addressed through matching, not through inverse probability weighting, I understand.
That is one method as well in combination,
but in this design they did matching.
Yeah,
I understand correctly.
Yeah, in this design, I have to say we didn't go very much.
May I probably I think that probably is not needed, Rosa, because the follow-up will be quite short. I mean, it's the time, the risk window for identified after vaccination. So probably the assumption is that baseline confounder does not change during follow-up. That is, I think, if I'm not wrong, it's 45 days maybe for GBS.
No, I know. But what I see more recently is that they don't do matching for variables. They prefer, because this would exclude, would exclude cases. They would rather wait through inverse probability. That's new. I mean, I haven't seen in recent studies. I've not seen this matching. That's why. And then
I see.
So this is for confounding, but then inclusion criteria. Okay, so confounding is only addressed through matching, right? Because the rest is for selection bias.
Yeah, through matching and other statistical methods, we can adjust for confounding after. I think to your point, one of the reasons they do matching is because it's so important to match. Because I'm no expert here, I'm just presenting their work, but I think that they want to match, that matching the gestational week is extremely important. Like I said, the timing, the exposure window. So in a pregnancy study as such, especially with this risk of preterm birth, which it'll come up in later slides, and maybe that will be more clear why it's so important.
Okay, thank you.
If that helps.
Thanks so much, Samantha.
Yeah. Okay, so the study period considerations. So the first maternal vaccine, as I said, was approved in 2023. So the authors propose starting data collection from 2022 to allow for a 1-year look-back period to capture prior medical history, comorbidities, medication use, all critical factors for an accurate analysis, especially with when studies of causal inference. And the data collection will be until the last data availability for each database. And given the anticipated need for a significant number of patients, and that we don't know about the vaccine uptake, at the moment they recommend a 5-year follow-up period. So then, just in case for those of us who are not as familiar with the Target Trial Emulation framework, it's an analytical framework developed by Hernan and colleagues, and it's using observational research to mimic the design elements of a randomized controlled trial using real-world data. It's become increasingly popular in clinical research, especially when running an actual RCT isn't practical or too costly. And you can see in the bottom 2 boxes here that there's 2 stages of the framework that they highlight, and that's that stage 1 is specification of the target trial, where we formulate the causal question as if we were writing a randomized trial protocol. So we define the causal estimands and the analysis plan, and set up the ideal trial that we want to emulate. Then stage 2 is emulation of the trial using observational data. So we identify eligible participants, assign treatment strategies that align with the protocol, and follow them from time 0 or until the outcome, from time 0 to the outcome or till the end of follow-up, and analyze the data as if it were a target trial, but then you'd adjust for baseline confounders to approximate randomization. And we're going to go through in the next slides each of these steps. Oop, wrong way. So this is an example of a target trial specification table that compares the target trial to the emulated trial. I know there's a lot of text here, so I highlighted the differences in red. And I know we said we would save questions till the end, but I did want to first see if anyone has any idea what challenges might arise when trying to set eligibility criteria and the emulated trial when using observational data, no pressure. If not, we can talk through them here, but just thought I would open the floor up so that it's not just me talking the whole time.
Yes. Hi, can I make a comment? I'm Giorgio. Yeah. Yes. So I see that the target trial that they are trying to emulate is the MATIS, right? The target trial that is already done. But in my opinion, should be the MATIS trial. We are not trying to benchmark this clinical trial, right? In this paper.
When you say
benchmark, sorry, let me motivate it. I've been going through articles in which they benchmark a randomized clinical trial. They take a clinical trial and they emulate it following strictly the specification of the clinical trial, and that is like used in order to prove the robustness of the target trial emulation. They compare the results and they hope they are similar. But in this case, I think the purpose of this protocol is to extend and overcome the limitation of the randomized trial MATIS. Am I right?
Correct. So we take that. I mean, we take this criteria and then, like, for example, we maintain some of the criteria, but then we make it less strict. For example, we don't want to include or exclude people with complications or put age restrictions, but we want to maintain the 24 to 30 weeks gestation requirement because there's also a lot of uncertainty around the safety and the risk of preterm birth. Does that address the question? Did I understand correctly?
Yeah. I just wanted to say that maybe I would specify an hypothetical target trial that is different from MATIS and that overcome the limitation of MATIS instead of using MATIS target trial as the hypothetical target trial that.
Yeah, and I suppose you could use multiple clinical. Are you saying that if you use multiple clinical trials, and then you, from there, you create your target trial that you want to then emulate? That's kind of first looking at that and then emulating once you have that target trial you want. So for the purpose of this study, I believe they were using the MATIS.
Okay, yes, thank you.
Yeah.
I saw that this hand raised.
No, yes. it was an agreement with you. I think it creates confusion if you want to. It's different if you want to emulate a trial to prove and keep as strict as possible to prove that your results are aligned with the target trial is one thing. But I think this is not what we are doing here. And then I agree, Giorgio, that it would be clearer, neater somehow to design an hypothetical trial, not the MATIS, that is very similar to that addresses the same research question that we are addressing in the emulation, because clearly MATIS was addressing a different question.
Yes, exactly. I saw the paper they're using this MATIS as hypothetical target trial, and yes, that was my. Thank you.
Do you have a comment? Nick?
Oh yeah, hello. Hi, thanks. Yeah. Well, you were just asking about some of the issues here. I mean,
one of them might be when you're trying to do the emulation. I mean, you're assuming that all the information that you need is available at the time that you want to do this this randomization. So you need to make sure that because if you're basing this on, I don't know, a cohort data set, it might be that some of the information you're using is future information. So you've got to be quite careful about that. And I think it's sometimes quite hard to know whether that information is future information, for example, about risk groups and so on. And that's one thing. And the other thing is, one of the big issues is, I mean, do we know whether the individuals, whether the pregnant woman is, for example, not in hospital waiting to deliver, or having a complication, meaning they're in hospital where they're unlikely to actually get a vaccine? Because you know, in your target trial, they almost certainly wouldn't be recruited in, but in your emulation, do you know that at that time that they're actually not already in hospital and likely not to get a vaccine, for example? So there's things like that which are potentially difficult to emulate and can cause bias in your target trial analysis if you don't think about these things. Over.
Yeah, I completely agree. That's a good point. It's when working with the data, like the feasibility of the data sources, and also knowing it's really hard to know what you're going to be able to, what information you're going to have. And then, as we were saying before, the timing is really important for this. And that's also something we touch upon a bit later. So thank you for bringing that up. So then I'll go ahead and continue with this table that has the eligibility criteria, treatment strategies, and the assignment procedures. So as I already mentioned, that the age is something that we removed for the criteria, and then we kept the weeks of gestation, and we stratified the high-risk pregnancies instead of excluding them. Then for the treatment strategy, it's similar to the emulated trial. The main difference is that we don't have a placebo, and then for the assignment procedures in the target trial, women are randomly assigned to a vaccine or placebo without knowing which one they have, whereas in the emulation, we match exposed women based on the gestational age, calendar time, maternal age, immunocompromised status, and high-risk pregnancies. Now we can go ahead and go to the follow-up period. Oh, we're frozen here. Okay. Again, you see the red text highlights some of the differences. So in the target trial for maternal participants, the follow-up begins at informed consent and continues through 1 month after injection for adverse events and up to 6 months for serious adverse events. And we see the differences here in the 2 columns. In the emulated trial, follow-up starts on the day the eligibility criteria are met and ends at the earliest of however many months after birth, depending on the outcome of interest, maternal death, disenrollment, or migration, data availability limits, treatment crossover, or occurrence of the outcome. And then finally, we have the outcomes, causal contrast of interest, and analysis in the target trial versus the emulated trial. So for the target trial, for safety, the primary endpoints included reactogenicity, hard word, adverse events in maternal participants, and newly diagnosed chronic conditions in infants. And the emulated study focused solely on safety, so that's also a difference. They have the efficacy endpoints here. And for the causal contrast, we look at the per protocol analysis approach in the target trial, which means the estimated effect among those who adhere to their assigned treatment. In observational studies, we then have the observational analog per protocol effect. And what this means is we create an analog of these effects using real-world data and statistical adjustments. We restrict the analyses to those individuals who meet the protocol-like conditions and adjust for baseline confounders to emulate randomization. This approach is only restricted to pregnancies without a loss to follow-up for the study. And it allows us to frame the causal question as if we were running a trial and we make transparent assumptions about the trade-offs compared to the ideal randomized, ideal RCT. Then this slide touches upon the data source feasibility. So the authors highlighted some criteria that are really essential for ensuring our data sources are reliable for robust causal inference. We need data that can accurately determine the pregnancy start and end dates and detect ongoing pregnancies. It should also allow us to identify the RSV vaccine exposure and timing and enable mother-to-child linkage. We should be able to capture the relevant outcomes and confounders, especially important. And the data should represent a population with well thought out, thorough demographic sampling and include detailed individual level healthcare information, which can be quite challenging. And it must support patient monitoring over time with accurate recording and standardized coding to reduce misclassification and mitigate other biases. Oops, apologies, went too fast forward. So one of the most important and challenging elements in emulating a trial is determining the time zero. In a hypothetical RCT, the follow-up would begin immediately after following assignment to the vaccination or the controlled group. In an observational study, the timing of the vaccination and initiation of follow-up may not temporally align. So that means that individuals' vaccination status can be unexposed for part of the pregnancy and later exposed for the remainder of the pregnancy. If the vaccine exposure is classified as a binary variable based on the final vaccination status at the time of delivery, the period of pregnancy prior to the vaccination becomes immortal because they had an adverse pregnancy outcome. It would have been attributed to the vaccinated group or the unvaccinated group, apologies. And we're going to continue this discussion in the following slides. But since the opportunity to be vaccinated during pregnancy increases with the length of gestation, those who deliver at term have more opportunity to be vaccinated than those delivering earlier, making vaccination appear more protective of earlier events. For example, with the concern of preterm birth. So this slide shows the matching strategy in a target trial versus in the cohort study, and also touches upon the issue of treatment crossover, kind of what the previous slide was leading up to. So in the target trial, we see patients are randomized to either receive the vaccine or the placebo here, and they're both followed up for their outcomes. But then in this observational down here in the lower figure, study instead of randomization, they're matched based on their gestational age, calendar time, and maternal age. And then we also see that one person here could have the RSV vaccine and no vaccine. And then maybe later this person that didn't have the vaccine, they have a treatment crossover, and they do get the vaccine. And this, I think this slide is going to be helpful to keep in mind for the coming slides. So we're going to discuss what happens when patients change treatment groups, and how that's handled in the methods that the authors propose. So, but when this happens, a patient, for example, just to go back to that point, the patient would be censored from this no vaccine group, and then they would go into the vaccine group.
Rosa, do you have a comment?
Yes.
Oh, sorry,
yes,
I don't see everyone's.
I think it's an important comment. So you
Yeah.
are saying that in the trial, in the target trial, randomization would happen at conception, not at 24 weeks?
No, at the time when they receive the vaccine.
Well, that's only for vaccinated. But I mean, in the
Yeah.
design of the trial,
Right.
you would randomize not at conception, but at 24 weeks, right?
Yes.
Okay.
Between
so because
24 to 36 weeks. Apologies if I didn't
exactly.
phrase that well. Trying to look back. No,
so like I think
yeah.
that you would kind of emulate a trial every week or something like that, so that those how is that called, repeated trial? I don't remember now the word, but for sure not sequential, right? But
yeah.
for sure not at conception, because that would open a whole set of other
Correct.
problems, right?
Yeah. So are you talking about the actual trial, or like the target trial,
even
or the
actual
emulated trial?
Both, because of course you cannot know, even in a true trial, you cannot know when conception will happen.
No.
And you cannot.
No, but the vaccine is also recommended between 24 and 36 weeks. So I don't know if I accidentally said at conception. That's not what I meant.
No,
so,
in the previous slide you wrote T0 is conception, so I got scared. I said, no, come on. The last word.
I see. Okay. I see. Okay, I see your point.
Yeah.
No, that's an important one indeed.
That's very important, because otherwise it's a wow. Thank you.
Yeah, of course. So another analytical concern is the gestational age at time 0, which directly impacts the risk of preterm birth. So it's imperative to make sure that there's a robust, as we've mentioned many times, on the gestational age. One way of doing this that the authors recommend is, I think you were leading to that before, Rosa, was the emulating a series of separate trials for just different gestational ages at vaccination. Or it's also referred to as a sequential target trial emulation. You have the week 1 where you assess eligibility, and then do they receive the vaccine or not? And then there's a time 0 for that trial, and then they're followed up. And then again, we assess eligibility, and this is where maybe we have some, again, treatment crossover. So then those patients that were who are not vaccinated, maybe they're censored at the next week, and then they go into the vaccine group, and then they're also followed up. Go on to the next slide. And then just more on this idea of the series of trials, the sequential target trial emulation approach. It helps us address a lot of challenges. So this approach mimics, analyzes data in a way that kind of mimics a series of small trials, which each represent a different time point when a treatment decision could be made. And the trial emulations start at different time points and have its own eligibility assessment and treatment assignment and follow-up period. And this is a particularly appropriate approach when treatment decisions can occur at multiple points, or when eligibility can be met at multiple time points, or to address any time-related biases. In the case of this study, it's helpful for also addressing the distribution of the gestational age at time 0, addresses the issue of the treatment crossover, and aligning time 0, because there could be multiple time points when a patient could be eligible. Oh, yeah, I wonder about that. Okay, this slide addresses the statistical considerations. I see we're kind of getting a bit over the time that I was allocated for presenting, so we'll kind of go a little bit faster for these ones. But basically, the main statistical considerations for this study size, the authors recommend including all eligible women during the observation period. The outcomes, the measure of the different, depending on how common the outcome is, we use different measures. Power calculations are stratified by outcome frequency, and different approaches for common versus rare outcomes For preterm birth, they recommend applying specific analysis strategies to increase statistical power. And then other considerations which they highlight is the considerations for selective censoring using inverse probability weights, handling treatment crossover and censoring bias, and then exploring effects of censoring and the impact of excluding medically iatrogenic live births with a sensitivity analysis. So I guess the strengths and weaknesses of this methodology for assessing RSV safety in pregnant women. The protocol uses a Target Trial Emulation framework, which helps reduce bias from censoring immortal time and competing events and confounding. This also improves interpretability of the causal estimates and aligns observational studies with randomized trial participants or principles. Apologies. It also, the sequential or the series of weekly trials matching participants by gestational age of vaccination introduces a continuous outcome time between vaccination and birth. This offers more statistical power and mechanistic insight. But despite the strength, there are many challenges that still persist. So there's a risk of outcome misclassification, especially for preterm birth, as this needs validation to distinguish spontaneous from medically indicated cases. Exposure misclassification is another concern, as RSV uptake may not be fully captured in the current data sources. Maternal immunization practices vary widely across countries, and data sources may lack granularity for some covariates. And that's as fast as I can wrap it up. I know I went over the time, so I apologize for that. But also part of the rest of the session is just to, if anyone also wants to think of any other challenges that I didn't mention or propose any solutions to these challenges or has questions, then that's what we have the last 20 minutes for.